Assalamu alaikum guys, hope you are fine. Today I am discussing about the topic counter-current hypothesis. So what is counter-current hypothesis? This counter-current hypothesis is the continuous process of formation of concentrated urine. So let's discuss about how this process occurs. For better understanding, we will divide this process into several steps. The first step, we consider that the loop of Henle is filled with fluid having concentration of 300 milliosmol per liter. The fluid of loop of Henle having concentration of 300 milliosmol per liter, and this time the fluid in the medullary interstitium is also 300 milliosmol per liter. This is the first step. The second step, this is the descending loop of Henle and this is the ascending loop of Henle. We know the ascending loop of Henle. In the ascending loop of Henle there are some carrier proteins which are responsible for the active sodium pump. These carrier proteins are responsible for active sodium pump. So what happens if the active sodium pump occurs? Due to active sodium pump, the sodium ions are pumped into the medullary interstitium. As a result, the osmotic pressure or osmotic concentration in the ascending loop of Henle decrease and reaches 200 milliosmol per liter. And the osmotic concentration in the medullary interstitium or medullary interstitial fluid becomes 400 milliosmol per liter. This means a concentration gradient 400 minus 200 is equal to 200 milliosmol per liter. This sodium pump becomes active until this osmotic gradient is achieved. This is the second step. The third step, here we see that the osmotic concentration in the medullary interstitium is 400 milliosmol per liter, but the descending loop of Henle there is concentration 300 milliosmol per liter. So we see that there is an osmotic gradient also, and this is 100 milliosmol per liter. And we also know that in the descending loop of Henle is very highly water permeable. As the descending loop of Henle is highly water permeable and a concentration gradient is arise, so water now moves from the descending loop of the Henle to the medullary interstitium. Here we show that water moves to the medullary interstitium. When the water moves to the medullary interstitium, the osmotic pressure in the osmotic concentration in the descending loop of Henle increase, but the medullary interstitium osmotic concentration should decrease. But in this case, the active ion pump in the ascending loop continues. So the medullary interstitium pressure osmotic concentration remains same, 400 milliosmol per liter. This is the third step. Then the fourth step occurs. This is the step 4. So what happens in this step? In this step, new fluid comes to the loop of Henle. So this fluid, we consider there is a fluid. So now this fluid will come to the ascending limb of the loop of Henle. So this portion of the fluid is now reached this portion. So this part of the fluid becomes 300 milliosmol per liter because it comes from the proximal convoluted tubule. And the osmotic pressure in the lower part of the medullary interstitium 400 milliosmol per liter, and the new medullary interstitium fluid হচ্ছে 300 milliosmol per liter. And this 200 milliosmol per liter concentration fluid reaches the position. So then occurs the fifth step. In the fifth step, the active sodium pump again occurs, and the fluid of the ascending loop of Henle, this concentration reaches the 300 milliosmol per liter from 400 milliosmol per liter, and the medullary interstitium osmotic concentration becomes 400 to 500 milliosmol per liter. And 200 milliosmol per liter concentration fluid now becomes 150 milliosmol per liter concentration, and the 400 osmotic pressure becomes the 350 osmotic pressure. Here the osmotic concentration should be 300 milliosmol per liter. This is the fifth step. Then the sixth step occurs. In the similar way of the third step, fluid osmosis or fluid comes to the medullary interstitium from the descending loop of Henle, and the osmotic concentration become 400 milliosmol per liter, 300 milliosmol per liter respectively, and the medullary interstitial osmotic concentration become 500 and 350 milliosmol per liter concentration respectively. This process continuously repeated again and again until the concentration reaches at the tip of the loop of Henle 1200 milliosmol per liter. This is the maximum concentrating ability of the nephron. Thereby, or thus, the concentration of the urine increased, and this process is called the countercurrent hypothesis. I hope you understand the topics countercurrent hypothesis and how the concentrated urine is formed.